My name is Sungmi Lee. I'm postdoctoral researcher from Edinburgh University. Today I talk about plasma microRNA profiling study in thoroughbreds, racehorses, with stress fracture or without stress fracture, which is confirmed by nuclear scintigraphy. Co-author of this study is Eugenio Cillian Garcia, Mike Clinton, and Sarah Taylor. And thank you for other collaborator and our founder, HBLV. Catastrophic musculoskeletal injuries are important due to economic loss in race industries as well as welfare issues. Incidence of the catastrophic musculoskeletal injuries showed 1.17 cases per 1,000 starts. Among them, bone injuries are most frequent type of injuries. Non-fatal stress fractures are most common type of fractures. Stress fractures are incomplete fracture, which are associated with catastrophic fracture if not identified and managed properly. Stress fractures are a result of continuous stress-related bone injury during race training and racing. Repetitive loading in cortical bone causes formation of microcrack, which lead intense remodeling, and increase cortical bone porosity can decrease stiffness and strength in bone. If imbalance of remodeling continues, microcrack continues and form macrocrack, stress fracture. Since the stress fractures are high risk of developing catastrophic fractures, early diagnosis is important for interventional treatment. On the left side is a micro-CT shows microcracks in 3-year-old thoroughbred. Conventional imaging using radiography and ultrasonography are limited to detect stress fracture. Current gold standard diagnosis by nuclear scintigraphy, as shown in the right two images. In this study, we focus on microRNA, which has been considered as potential biomarker in many diseases. MicroRNAs are small non-coding RNAs. On the left top, primary microRNA from DNA is transcribed by RNA polymerase II into primary microRNA, and these cleaved by RNA3 enzyme into pre-microRNA in the nucleus. Pre-microRNA is exported to the cytoplasm, and the pre-microRNA is cleaved into microRNA duplex. One includes mature guide RNA, and the other one complementary passenger microRNA. After separation, complementary microRNA strand degraded, and the other mature microRNA incorporated risk RNA-induced silencing complex, which interact with the specific site of the mRNA and result in inhibition of translation or mRNA degradation directly. It has been clear that expression level of certain microRNAs are altered in many diseases. And also microRNAs are present in biofluids like plasma and urine, blood, and synovial fluid. This allows relatively non-invasive sample collection, and also it is very stable as form as exosome. Therefore, microRNA has been considered as a potential biomarker. Here are our questions. Can we see any difference of microRNA in bloods between stress fracture and two control, including lame control and healthy control, non-lame control? Once thoroughbred horses are referred in hospital with lame, we collected blood, and the stress fracture were diagnosed with by nuclear scintigraphy. In parallel, no lame control, healthy control were collected from yard horses, which are also thoroughbred and underwent a similar training and environment. Plasma was separated and used for the microRNA profiling by small RNA sequencing. Small RNA sequencing is a technique to identify all kinds of RNA molecule, small RNA molecule to identify microRNA profiling and to understand the role of the RNA. Our profiling study included 5 samples from each group, and they are all thoroughbred and horses the similar age around 5. The location of lame was mainly in metacarpal or metatarsal bone, and stress fracture included 2 tibial fracture and 1 MT3 and 2 MC3 fractures. After sequencing done, the quality of small RNA libraries were evaluated. This is one representative summary of the sample. This is to identify accuracy of identified nuclear base by sequencing. Fred scores above high 30 corresponded to 99.9% of base calling accuracy. A shows average of Fred's quality per base is high, above 30, suggesting high quality sequencing done across all bases. We also showed the average quality of most sequences was high, with score above 34, and C showed the distribution of read ranks after removing library and sequence adapter. The abundant peak of the dispersion is around size 22, indicating abundance of microRNA peak. Finally, we obtained average 6.6 million reads, and the map percentage was 75.2%. Clustering data set is to explore the data set by reducing many variables, in this case, expression of microRNA. And so in the smaller one, while preserving as much of information as possible. This allows to separation of sample in the different region of PCA plots corresponding to their biology. In here, the reds, the sample between the sample is close together, but there is a close pattern of microRNA expression profiling between stress fracture and lame control, red and green, which indicate the 2 microRNA profiling has similar microRNA profiling. But the blue, the healthy control, no lame control, was separate from the other 2 groups. Differentially expressed microRNA was investigated. Our interest was to distinguish stress fracture from 2 control, so we combined one disease, the lame control, and non-lame control, healthy control. After normalization using scaling factor, microRNA 211 was only microRNA identified as at a statistically significant level of differentially expressed microRNA. It was downregulated and in stress fracture with fold change 0.44. And also there was a similarity of microRNA profiling between stress fracture and lame control without stress fracture, which was also consistent to previous PCA observation. How about the lame control versus healthy control? There were much more differentially expressed microRNA when lame groups were combined. These microRNA show top differentially expressed microRNA. One of most abundant microRNA, 486.5P, was downregulated in lame groups, and the other microRNAs 21, 23, 26A, 148A, and 103A were upregulated in lame groups. These results show microexpression pattern of lame cases differ from non-lame healthy control. We also validate RNA sequencing study by qPCR. The first graph showed the quality check, using the external spiking control of cell mirror. All cycling threshold, the level of expression of external spiking control were similar level. First, microRNA 211 was not validated by qPCR to distinguish stress fracture from control. Unfortunately, in this case, the plasma, the level expression of the microRNA was quite low and used with high cycling threshold. Maybe it might require further validation. differentially express microRNA between lame and non-lame microRNA 4865P and 23 and 26A were validated by qPCR. To summarize, this is the first study of microRNA profiling study in stress fracture. First of all, microRNA expression pattern of lame cases combining stress fracture and non-fractured lame differ from non-lame healthy control, which indicate microRNA profiling reflect the biological differences. However, there was a similarity of microRNA profiling pattern between stress fracture and lame control without stress fracture. Due to the study design, the result might be only claim association, not causation. Further evaluation of microRNA 211 in a large cohort of cases and in vitro are required. Next plan is to validate microRNA expression and the role of microRNA in vitro using horse-derived cell, for example, MSC or chondrocytes. Thank you for the listening.